Okay, this is the first unit on the nervous system. So our nervous system is composed of two parts. We have the CNS, which is your central nervous system, that's going to include the brain and the spinal cord. These structures are all encased in bone. The PNS is your peripheral nervous system, and this is going to be the branches that come off of the spinal cord, as well as a few of the cranial nerves. Your nervous system has three major functions. You've got the sensory functions that are going to perceive the stimuli, the motor functions that are going to carry out responses or send messages. You can think of the sensory functions as being information coming from the body to the brain, the motor functions coming from the brain to the body, and then the integrative functions that are going to process information from the body and determine the appropriate response to carry out. Most of your integrative functions are going to be in the CNS, largely in the brain, but some of them will occur in the brainstem and spinal cord. So this outlines our main organization. You've got the two main branches, the CNS and the PNS, and then within the PNS, we've got the SNS, or somatic nervous system. This is going to be to the body. It's the things that are under voluntary control and things you have conscious awareness of. The ANS, or autonomic nervous system, is going to be divided into two modes. It's going to operate either under the sympathetic, which is your fight or flight mode, or your parasympathetic mode, which is the rest and digest. A lot of these you do not have conscious control over. You may be able to consciously change some things in your environment that will influence these, but you don't have direct conscious control over these. So when we look at the neuron, the cell body is going to be the portion of it that's going to contain the nucleus. It's also going to contain your genetic information. The dendrite is going to be the receiving end. On this diagram here, it's showing multiple dendrites where it can receive information. The axon is the sending end. Sometimes you'll see it referred to as the dendrite end is going to carry messages towards the cell body. And the dendrite is going to carry messages away from the cell body or send them. And then you've got the axon terminals of the dendrite end. The synapse is the site of communication. This is going to be where it either joins with the next neuron that it's going to communicate with, or whatever its target is. It's that place where the nerve connects to it. Neurons are pretty delicate and are not replaced well in the body, so we've got the neuroglia, which are your support cells that are going to help to nourish and protect your neurons. Their main interest is to make sure that the neurons are not damaged. Some of your neurons are going to have a myelin sheath, which is a fatty protective sheath made of the fatty acid DHA around it. And that's what you're seeing right here, is that myelin sheath in there. And DHA we can acquire from the diet, but the biggest source of it in the human diet is usually going to be EPA oil, eicosapentaoic acid, which is going to be easily obtained through eating cold water fatty fish. If you don't eat fish, there are other sources of getting DHA into your diet. And then the nodes of Ranvier are going to be the spaces in between the myelin here. So with our nerve tissue, the Schwann cell is going to be what actually forms the myelin sheath in the PNS. A lot of times you're going to have structures that will exist in both the PNS and the CNS, but they'll get different names. So the Schwann cell is one example of those that's going to be in the PNS. The neurolemma is the thin sheath around a nerve cell. We've got several other support cells in there. The astrocytes are a star-shaped cell that are going to provide physical and metabolic support of the neurons. The microglia are the immune cells of the nervous system, and then the oligodendrocytes are going to myelinate the CNS cells. So here you've got the Schwann cell in the PNS, oligodendrocyte in the CNS. So this is showing a cross-section of the brain here, showing your white matter and gray matter. Gray matter is going to be unmyelinated, and white matter is going to be myelinated. They're not actually white and gray, they're actually two different shades of beige in there. Your white matter, with having the myelin on there, is going to conduct impulses faster in there. It will conduct impulses through what we refer to as saltatory conduction, which is going to be faster. It tends to jump over the spaces. It only has to depolarize in between the myelin. That can be quite quick. It can go up to 100 meters per second. The gray matter is going to do continuous conduction in there. It doesn't have the myelin in there, so it has to depolarize all along the membrane. That's going to be slower, although slow is considered up to about 10 meters per second in there. So both are quick, it's just one is very quick in there. So how we're going to conduct these impulses is we're going to establish two different sides of the membrane here. Through a sodium-potassium pump, you're going to have sodium being pumped to one side of the membrane and potassium being pumped to the other. For every 3 sodiums that are pumped out, you're going to pump 2 sodiums back in. This will establish a gradient where you're going to have lots of sodium on the outside and lots of potassium on the inside. When you open up one of these channels in here, what's going to happen is the sodium and potassium want to be in equal concentrations on both sides of the membrane, so the sodium is going to rush into the cell again there. So there is a link here that you can follow that's got an animation of the sodium-potassium pump that you can watch from your notes. The sodium-potassium pump is going to be what helps to establish our action potential in there. Across the membrane, we can measure the polarization in there. And when a membrane is polarized, we say it has membrane potential. It's got the potential to do work. By having the separation of the sodium and potassium and them wanting to come together, that membrane has the ability to do work by allowing them to come together. And we can measure how much that is. It actually creates an electrochemical gradient, and it's negative 70 millivolts. So that can be measured with a voltmeter, just like you would measure on a battery. On a battery, you've got positive and negative charges separated, and on this membrane, you've got the sodium and potassium charges separated. So this is an animation of this occurring right here that I would strongly recommend you watch as well. All of these can be accessed from your notes. The stimulus is going to be what changes the resting potential. It's going to be what triggers those gates to open and allow the sodium to come rushing back in. It's going to automatically want to rush back in, but what causes the gates to want to open is going to be your stimulus. So when we go to spread this action potential, you're going to reach a threshold, which is a critical level of depolarization. So you have to have enough depolarization for those charges to actually be able to come together. It operates on what we call the all or none principle. As long as there's enough depolarization to reach the threshold, the sodium-potassium gates are going to open and the action potential occurs. The physical peak is always the same. So the threshold is how much depolarization you need. Depolarization is the coming together of charges across the membrane. So they're never going to come just halfway across. It's either going to fully depolarize or not depolarize at all. Repolarization is the separating of charges across the membrane. So you can think of depolarization like when you have a battery in a toy and the toy is turned on, it's draining the battery. When you take those batteries out and you charge them back up again, you're repolarizing it. So here in depolarizing, the charges are coming together. With repolarizing, you're putting energy into it to separate the charges again. Sodium and potassium are coming together and depolarizing. In repolarizing, we're separating them again. So one of the things I want you to think about is if you have depolarization is always going to occur all the way, you have this all or none principle, what would cause you to have only part of a nerve signal be transmitted? So I want you to think about, why would you have numbness or tingling in there, or only part of the message getting through? If the action potential is truly sending the whole message, is truly firing off, what is causing that message not to get to the other end? And a little hint here, think of the last unit that we covered. And here we have another animation here that's going to show the physiological events and the neural synapses. So when we look at synaptic transmission between neurons, you've got the presynaptic neuron. It's going to be the neuron delivering the stimulus to the synapse. The postsynaptic neuron is going to be the second one that receives the stimulus from the synapse. Whether or not you're presynaptic or the postsynaptic neuron is going to depend on the relationship. You could be a presynaptic neuron in one relationship and postsynaptic in another. So if you have neuron A delivering a message to neuron B, neuron A is going to be the presynaptic neuron, neuron B is the postsynaptic neuron. If we look at the relationship between neuron B and neuron C, if neuron B delivers to neuron C, neuron B is going to be presynaptic in that relationship with neuron C. So what will happen is the impulse is going to travel down the presynaptic end of the presynaptic neuron. This is very similar to the synaptic transmission that we looked at the neuromuscular junction. It's going to depolarize, and when it gets to the end of the neuron, neurotransmitter is going to be released. There's various different neurotransmitters that are involved in the nervous system. At the neuromuscular junction, what we looked at was only using acetylcholine. Acetylcholine is one of them, but there are other neurotransmitters which we'll look at in subsequent units. And then the postsynaptic neuron is going to depolarize. So again, here we have another animation that is going to show this process. So that is the end of the outline for this first section of notes. I strongly recommend you watch the animations. A lot of times these concepts sink in a lot better when you actually get to see them happening, and that's why those animations are so useful.